Good morning, everyone. And today we will discuss about CRISPR Cas system. So while we are actually concerned only with the applications, as because this is an applied course in recombinant DNA technology, and we are supposed to learn more of genome editing and other stuff. But it is a general idea. We have to develop a general idea about many things. It's not just it's an application, and at one point we all should be able to understand the actual biological origins or evolutionary origins of these. What are these systems actually doing? And the idea for me to take up this biology and mechanisms is to appreciate how nature comes up with ingenious genetic elements. And the most importantly, as a student of rDNA technology, we should be able to see how basic science can be manipulated to applied science, right? How can we translate the information that we get from basic science to applications? In this case, that is one of the reasons why I want to take up basic science. All the tools come from all the tools are coming from nature, right? What I mean nature is every genetic elements that are there in various different organisms. Say lambda phage, for example, everything, majority of them are we are using these tools, genetic tools that are coming. They were discovered and they were understood how they function, characterized. That there they were just trying to understand how it works. That's basic science. And once that is there, once we find and characterize, then we know what all it can be applied to, and then we will customize the naturally occurring genetic tools for a particular application. So it's important for us to also be in that. I think in lambda phage 2, we discussed about biology of lambda phage and then the applications. Same with case with M13 phages. So that is how we need to understand the importance of basic science too. So broadly, I would, this is a general overview, and the CRISPR Cas system is dubbed as the immune system of bacteria. Okay, they were identified some 50, 60 years ago that they found there are several regions like this with some spacers, and then there is a palindromic repeat like this. And at that time, they didn't know what it is. First, they first identified, and then they saw thought there are repeats like these, and there is a space, definite amount of space between them in terms of bases. And why do they have it, and what is their significance? They didn't know. But with progressing research and again revisiting what it was found a long time ago, now we know many things as now we know that as CRISPR. It stands for clustered, regularly interspersed, short palindromic repeats. So this is CRISPR, and so we will also just look at what other proteins are there. And there is Cas, right? CRISPR associated. That is CCAS. CRISPR comes from the first words in here, the acronym. And then there is also one more sequence called as PAM sequence, protospacer adjacent motif. Instead of trying to mug up everything just as it is, if we can associate what you mean by these, then it's easier to remember. Clustered, you will find many of these repeats like this, and they're all present in one place. So it is clustered and regularly interspersed. The space I was telling you is little, there is a definite space between them, and then you're finding these are the repeats. Okay, there is a space here in between, and there are repeats. So clustered, regularly interspersed, that is about the space. Short palindromic repeats. These are the palindromes. What is a palindrome? Like we learned in restriction endonucleases, you read from here and here, the sequence is similar, right? So they are clustered, regularly interspersed, short palindromic repeats. And there are also other genes encoding proteins, which are CRISPR-associated proteins. That is where the Cas comes from. Okay, there are four important stages. First thing is, as we said, they call it as the immune system of E. coli. And what is an immune system in our bodies? Our body's immune system will eliminate foreign objects or proteins, you can say, or molecules. We'll just call it as molecules. And not just how, that means they have a way of determining what is self and what is not self and what is foreign, you can say, right? They should be able to recognize which one is foreign, which one is self. Only then they will be able to eliminate the foreign. Second thing is about immune system is it has memory. That means if the immune system has encountered a foreign object, then within the body they would have attacked it, and then they would have stored that this is a kind of this foreign object has entered into the body, and they will have bigger response or made fine tune the immune system for that, because there is a probability of that foreign object entering it again. So how swiftly can we eliminate, how swiftly and efficiently can we eliminate it is important. So I also like to, I think I might have discussed, but I will also tell you this. I like to go to web pages of different scientists, and they have their own pictures. They would also have their, they have nice images sometimes made, which are not published. And they would also have listed out their research interests, right? If you are looking at for your graduate studies, or say semester abroad program or anything in the future, PhD or postdoc, it's always important to try to learn about their work. Then visit the web page, try to read it completely from start to the end. Then you'll get better ideas because you know their objectives. Then, and probably you know how they're trying to address also. Many labs abroad, at least, they do give a good understanding of what their lab is happening. So we should make it a habit to go and try to read more on those web pages. It'll give a quick overview of many things those people in that lab are doing. Okay, so there are four steps in this in the in with CRISPR, and the four steps are adaptation, right? Okay, before going, I will again repeat what it means by the immune system. To be able to distinguish from self and foreign. Okay, you have to remember this until we finish today's class, that they have this self and foreign, and then there should be memory. Only then we can qualify it as immune system. So the steps included are adaptation. We will go through these steps again, but I'm just giving an overview here to describe what it means. And then we have this second step is expression. Third step is interference. And fourth step is priming. Say, not all bacteria have a CRISPR-Cas. Okay, these systems are not present in all bacteria. There are in some strains, some E. coli strains, for example, do not have it. Adaptation is about generating memory. Okay, that means if a DNA from a phage has entered into the bacteria, say lambda phage has injected DNA into the, I'm not showing, Lambda phage, for example, some lambda phage has injected the DNA, the red one that is shown here. And so what happens is there are Cas proteins, some different types of Cas proteins are also present. These Cas proteins would have cleaved this DNA. Okay, it's not that always it happens successfully. Sometimes it's successful, sometimes it's not. So in those bacteria that the cleavage of it has been successful, those regions, these small bits, would be taken and would have been incorporated here. Okay, so it is making a memory of a phage DNA within itself. And now the spacer has been integrated. Now you have the red one that is shown here, that is the spacer integrated. From the Cas genes, we will get Cas proteins, and then from the CRISPR genes, you can say CRISPR elements, you will get the mRNA, and which can be folded like this because it's a palindrome, we said. And it will be further processed to generate CRISPR RNA like this. So each of them will have a protospacer or a spacer, and then you will have the palindromic region, the hairpin loop. So that is about expression. The third is, say, for example, a similar um, phage again, I mean, after several generations of E. coli, I mean, cell division, if it has attacked, okay, if the DNA from here, say, yeah, this is the second time the infection has happened, the DNA is injected, but we already have a spacer for it, right? So that would come and sorry, here, yeah, you can. This is a surveillance complex. Let me take back again. CRISPR RNA, then it binds with the Cas proteins. There are different Cas proteins. And if the phage has infected, it will bind to the CRISPR RNA along with this complex, will bind to the target based on complementarity, and then it will cleave the DNA, right? This is the DNA from the phage. It is attached by the CRISPR RNA, and once it is attached, the several regions are broken down so that the lambda phage cannot establish any lytic cycle or lysogenic. And the fragments that have formed may further make more spacers into the CRISPR, right? So that it will act as a memory. And next time if there is any further infection, it will have effective um, effective response against the phage. That's in a way overview. So the four steps we should say are the adaptation, expression, then we have interference, and then the priming. Now let's try to have a look at the molecular steps. And please remember that if we just discuss about CRISPR and all the ways that is characterized, it will take a lot of time and it's beyond scope. But I just want you to understand or actually appreciate and enjoy the way CRISPR works. That's more important than for me than the applications, because here this is just the natural selection has come up with something, and it is at the level of DNA or nucleic acids, and it is a fight between, you can think of bacteria and a phage fighting. And what are they fighting with? Using DNA, or they're actually fighting with in our immune system. The fight is between cells, right? And we fight. One nation fights the other nation. One community fights the other community. We have different things to do that. Even cells, one cell, one bacterium can fight with another and one organism that way. But what does it take for a phage? The um, fight between phage and bacterium. How is it mediated? It is mediated by molecules. And I mean not molecules like antibiotics. That is also another warfare. This is directly involving information, right? That is directly involving the genetic information. That's why I love this topic. And that's why I love this topic of trying to understand the biology. Okay, so the first things, we'll just look into it again. So locus and organization. So we have, as I discussed before, there are several repeats, and each of these repeats has spaces like this. Okay. And the spacers, what they do is, I mean, this is the CRISPR array. You have sequence of them, so it is an array. I will try to see clustered, regularly interspersed spacers, sorry, short palindromic repeats, right? It will um, it will get, we have to get used to it and keep using it often. Interspersed, this is regularly, and this is clustered. Clustered, regularly interspersed, short palindromic repeats. There is a leader, and you have a promoter here, which will allow the transcription of this whole region as one mRNA. Oh, sorry, not mRNA, that's RNA, and which will further be, um, we will have cutting and maturation of those. We'll come to that later. There are also other genes somewhere nearby. Those are called as CRISPR associated genes. Okay, Cas comes from CRISPR associated genes. Okay, that is the organization of the CRISPR array and associated genes. The second step is adaptation. So in the first instance of an invader, like we said, lambda phage, for example, has infected DNA, infected the bacterium, and injected its DNA into the cell. There are two proteins which are called as Cas1 and Cas2. They are the proteins that will bind to these foreign DNA, and they will bind to the DNA where there is a region called as PAM sequence. Okay, we'll get to that. So wherever they find that PAM sequence, they will bind and they will cleave that region, and it is now referred to as, this is the DNA region of the small fragment of the of the lambda DNA. Okay, and it is cut and processed, and it is now called as the protospacer. And that protospacer is put into the CRISPR array. So that step, you call it as protospacer integration. So we have repeats, and these regions, the spaces are unique in the sense all the repeats are similar, right? And this has been acquired at one point when previous infection may have happened. Now this is a new infection that is happening, so they put in this fragment is integrated into the DNA like this here. And we will also see, we will repeat each of these steps again and again, so that probably we'll have a better understanding in that way. So the protospacer has been acquired, and once upon transcription, we will have this whole array expressed, which will produce one long RNA like this. And that RNA is processed, will be cut here like this, generating CRISPR RNA. And that CRISPR RNA will be bound to Cas proteins. These are different Cas proteins compared to Cas1 and Cas2 that we discussed earlier. So now we have the formation of CRISPR RNA Cas ribonucleoproteins. Excuse me. Okay, now we have a complex that is similar to that of our natural killer cells in our body. Okay, analogous, not similar. I'm trying to compare it with that of the immune system. And then we have, what does this ribonuclear CRISPR RNA Cas ribonucleoprotein does? If there is a second invasion by the same phage, okay, and then the interference, the CRISPR RNA, because it has sequence homology with this region, it will bind to the region like that. So the function of this CRISPR RNA here is to provide specificity. Specificity in based on sequence. And the Cas proteins are the ones that will cleave. Okay, they will cleave the incoming DNA. So there is a nucleus, a protein domain. I mean, there are multiple proteins, of course, in different systems. We will discuss a little while. So it will recognize there and it will cleave this DNA. So the phage infection has been averted. Okay. So these are the overview of the steps. There are multiple types of CRISPR proteins, and you can also see there are two, or we will just discuss, there are class 1 and class 2. The difference is based on the PAM. What is PAM again? That is protospacer associated motif. Okay, PAM sequence. And there are two classes. In one class, the PAM sequence is on this side, and in another class, it is on the 3' side. Okay. So you can see that, wait a second. And well, there are 4, 1, 2, 3, 4, 5. As people kept on recognizing or discovering, they found more varieties of CRISPR, which work on similar grounds, except there are two different categories. One of them are DNA cleaving enzymes, or DNA cleaving RNPs, we will call them as ribonucleoproteins, the CRISPR RNA, and the proteins are the Cas. And the other category is CRISPR RNA. These are RNA pairing. Okay, they degrade the RNA molecule and not the DNA. So those two categories are big ones. And then you'll find different papers. And I think if you keep on going into the most recent ones, you will find even more sequences or types, actually. In different organisms, they have found different types. And why I just put up this slide is just in case people can change these or use different systems for different purposes. And that is one of the reasons why we also need to know what variations are present in here. Okay. Yes, let's go ahead. So first thing is, how is the DNA getting integrated? So here is a foreign DNA. Okay, this is the foreign DNA that is coming in. I hope it is clear enough. The foreign DNA is here, and it will be cut by, we discussed about two proteins, Cas1 and Cas2. Cas1 and Cas2 would recognize them, and then they would have processed it to generate about 33 base pair protospacer. Okay, and each of them would have had 3' OH like this. And they would cut the DNA by recognizing these sequences. These are the Cas1 and Cas2 protein complexes. They recognize this motif, They bind there, and then they cleave some 33 bases here. And so we would get, they would have cleaved here and here to generate the PAM associated, this one GC, and then you will have the sequence from the viral DNA or phage DNA. Now, the DNA that is the genes, the CRISPR array that is present here, these are the palindromic repeats. We said this palindrome means the region here is kind of complementary to this region. You can say it like this. So if there is, I'm just making up, GACAT, if there is CAGA, and then we will have, it would have had the opposite, the complementary strand would have been AGCAT, TCTGAT, GCT. So this sequence is ATGCT. So I hope you agree that this region is complementary to this region. So if I put it this way, okay, I will try to write A, G, C, A, T, and assume these are C, A, G, A. And next we have this region, which will be A, T, G, C, T. So it can form a hairpin loop like that, right? So it is, we are not sure whether they form hairpin loop or not. There are indications, this cruciform like DNA structures, whether they form or not is not visualized, but people do believe that they can form that. I mean, here we are discussing about the DNA. We are not discussing about RNA. If it were RNA, there is a high probability, because if it is produced as single-stranded transcript, then we would assume that it would folded and form a hairpin loop like that. So what happens, or the question is, how does this protospacer get incorporated here? Okay, if it gets incorporated, there are two things we need to understand. How does the CRISPR, this protospacer, get incorporated into the CRISPR array? First thing. Second thing is, how is it possible that this repeat again is made again? Okay, if you remember transposition, and if you remember target site duplication, then it's almost similar concept here too. Let's see. So what happens is the processed protospacer here, it has 3' OH on both the sides, and the protospacer would this 3' OH here would make a nucleophilic attack and cleave here. Okay, so it is now covalently bound to the one of the fragment here, right? And the other one, the other side of the 3' OH would make a cut here. So if I were to write, I'll write it, I will see if I can make sense out of it. Let's see, this is the DNA, and the cut, assume that this region here, this is the repeat. I'll just put it as like that. Okay, that is the repeat there. And now we are making a nucleophilic attack. First cut is made here, and second cut is made here. And now I will try to move these two. Okay, and this is coming as, okay, sorry. I will do it a little differently because I'm unable to move them as I wanted to. Let me try again. So I'm going to draw it in 4 different 3 pieces here. These are the adjacent regions, and I have another, I'm trying to put this repeat here. Assume they are joined like this. and this is another repeat. Okay. What I was telling you was there is a nucleophilic attack here and here. And now I need to, what will happen is these, sorry, what will happen now is that this region will move like this. Okay. And we have this RNA we have here that is going to come in, integrate here, something like this. This is the protospacer, not the RNA, DNA part. This is the protospacer that has been integrated. The red ones I marked were the repeats here. So now the repair systems will fill in the gaps. And on both the sides, we have these like this. A new protospacer has been incorporated and the repeat has been generated. These are the repeats. That is how this whole system works, incorporating the new regions into the DNA. Okay, that is how the protospacers are acquired. I'm just repeating here. So the cut is made here and another cut is made here, and then this protospacer is incorporated and these gaps are filled in. And what we have now is repeat one, and the same thing has been repeated here too, right? This is how exactly target site duplication would work in case of transposons. So this is the first time when integration, when a phage has, this is the first time, assume a novel phage has been infected, and CRISPR, the Cas1, Cas2 proteins have been successfully able to identify, degrade it, and then they would have incorporated. Now, this system or the bacteria now is immune to that particular phage. So this is the first time when CRISPR has acquired the genome, acquired the protospacer, you call it as naive acquisition. That's the first time the bacterium was experiencing that particular phage. And then we have the CRISPR array, and the same thing, basically, it is cut here and here, and then we have a repeat duplication happening. So we have the new spacer is here, we have a repeat has been duplicated. So the next time when this protospacer is expressed and it is processed, you will have the CRISPR RNA for that particular phage. If there has been another attack by the same phage, now the bacterium already has this protospacer using which it will identify and degrade the phage. If it has degraded, that means there are many more fragments that are formed, right, for the same phage. And then that can be taken up and put in here. This is called priming acquisition. This is the first fragment. Using that first fragment or the first protospacer, it obtains many more spacers, and they all get incorporated. Some of them will get incorporated, so that next time there is an attack, there will be effective response against this phage, right? Yeah. So the question here is, how does we until now what we discussed is kind of how is memory generated, right? That is the first thing. Now we have to also discuss how does this system distinguish what is self from what is non-self or foreign. Maybe I should write foreign. The DNA, in both the cases, it is DNA, and both the cases, they have the bases, nitrogenous bases are GATC. Then how do we separate? In our immune system, we have different epitopes. Epitopes are typically made up of several amino acid residues or other molecules, complex molecules. If they are found within our natural body, then they are self. If they're not found in it, anything that is not found in our natural body is considered foreign. How does the CRISPR figure out which one is foreign and which one is not? And the problem is both are DNA molecules and both have the same nitrogenous bases. Then what we have is the probability of sequence, right? Sequence of these bases within them could be different, but how often can we have very unique sequences? One thing we can put is, assume there is a phage that has already that has infected, because of which we have a protospacer in the CRISPR array, right? And it has expressed and produced a CRISPR RNA. And we are saying that next time there is an infection from a phage, this CRISPR RNA will bind here and will cause the degradation of this DNA. Then the question is, why would it not cleave the CRISPR RNA, that is CRISPR array itself, right? This CRISPR RNA has come from CRISPR genetic elements that are present here, array. So why, if it can bind here to the target sequence of phage DNA, why can't it bind here and degrade this region? And this is present on the genome. And if there are multiple double strand breaks, then the bacterium would die because the genome is degraded. If there is no genome, no organism, right? So the question here for us is simple. We have we have a CRISPR RNA, and it can bind to the target phages and degrade it. And then why is it not degrading the CRISPR RNA that is present in the CRISPR array itself? CRISPR gene or the genetic element, the spacer is the same. So for that, there are different theories, and different people have found different solutions, kind of thing. So if we have the difference between the phage DNA, the target phage DNA, and the CRISPR array here is CRISPR array is having spacers on either side, right? So I just focus on this one, otherwise we'll take a lot of time. If it is the target, this one, the phage DNA, it would have complementarity to the spacer region alone. It will not have complementarity to the 5' end of the CRISPR RNA. It will not have complementarity with the hairpin loop region, right? Then the Cas9 would bind here and target it. If it is in this region, I'm talking about CRISPR array. Why is CRISPR spacer in the CRISPR array? How is it protected? If in that case, the repeat will have base pairing here, spacer will have base pairing, and also the repeat here also will have some base pairing, which suggests that which means it will only happen if it is self DNA. In that case, the nuclease activity of Cas proteins will not be activated, because of which the self DNA is protected. Whereas that's not the case when it comes to the target phage DNA, the foreign DNA, because these regions would not have been base paired, and that is kind of trigger for the nucleases to cleave the target. Okay, that's a good system, and it is kind of evolution that works out these ways. It's not directed; it's a random process, and those that have allowed survival on or existence will remain, and the rest would have died. Okay, so the Cas9 protein, the one that is cleaving, has multiple domains. The 6 domains rec1, rec2, bridge helix, PAM interacting, and HNH and RUVC. So this is the region which is important for binding to the PAM sequence. And when we say anywhere it cleaves the CRISPR RNA, first of all, in the naive acquisition itself, it is acquired based on the PAM sequence, right? And then we would have the protospacer. The same thing would also have come into the CRISPR array as well. And so if a if a protein has to if a CRISPR RNA RNP should cleave a target site, then there should be PAM sequence at the target site followed by a spacer. Only then the CRISPR RNA would bind here and mediate the degradation of this region. Okay. So I hope you know about RUVC, which you might have learned in homologous recombination. Yes. So there are, this is the Cas operon. Here we have the CRISPR repeat or spacer assay, CRISPR array. And Cas9 is the one that we have that is the important for nuclease activity. Cas1, Cas2 are for adaptation, the naive acquisition. They are done in different CRISPR arrays. Some of the genes are present and some other varieties can also be present. There is also one more that we haven't discussed is tracer RNA. Okay, tracer RNA is one another small bit that is present. When the spacer upon transcription, the spacer CRISPR RNA co-maturation happens, meaning that we have a precursor CRISPR RNA, which is the long RNA with all the, which is the RNA produced from this whole array. And the tracer RNA, the red one that is indicated here, will bind to a certain region like this, and then it will be processed by RNase 3 to generate a fragment like this. So the red one indicated is the tracer RNA, that is the mature one, which there will be a lot of processing happening here, like this. So we will get two RNA components. We'll get two RNA components. One is the tracer RNA, which is base paired with the CRISPR RNA. Now it is fragmented. These are fragmented like this. And they are bound to Cas9 protein. That is a complete, you can say, CRISPR Cas RNP. Okay. And both these element, tracer RNA and CRISPR RNA, are important. CRISPR RNA is for specificity of the target, and it is actually sequence specificity. And tracer RNA is something important in a way for interaction of CRISPR to the Cas9. And Cas9 is the one that is responsible for cleavage. This is kind of important for binding to CRISPR RNA as well as to the for the activity of Cas9. So when there is a target site, the one that is shown here, there is PAM sequence here. So the CRISPR RNA will base pair, and when this base pairing is appropriate and not further base pairing, we will have an R loop formation, something like this opening. And then there are two cleavages that would have occurred. One is on either side. Both the strands, there is a double strand break. And then the target DNA is cleaved, right? So we have, for the activity, we need CRISPR RNA, tracer RNA, and the Cas9. So all this from the how it is useful to the bacterium we have learned, and people have then started looking at how can they be used. They are something like the restriction endonucleases, right? Why are restriction endonucleases so nice for us is because they can make double strand breaks. Why do we want double strand breaks? Because we want to recombine them and make new types of genetic elements. Only if we know that we can cut, and it's not random cuts. We don't want cutting everywhere. We want to be in control of our experiments. So they have to be sequence sequence specific nucleases. That is why restriction endonucleases are great. They were awarded Nobel Prize for the discovery of restriction endonucleases. The same thing, I guess you know that Carpenter and Doudna, professors, they received Nobel Prize for discovery of these CRISPR. So CRISPR is also similar, right? What is it? What did we say about restriction endonucleases? They are sequence-specific nucleases. So we know where to make cuts, and so that we can make rDNA, recombinant DNA. You can recombine different DNA molecules. CRISPR is also the same thing. You have sequence specific, and that is coming from the CRISPR, and the Cas9 is able to cleave it, right? It is a little more complex than what restriction endonucleases are, but but CRISPR will give us flexibility. If you remember when we were discussing, sorry, yeah, if you remember when we were discussing about restriction endonucleases, say for example, there is a gene, and I want to, this is a restriction site, and here is a restriction site, and I don't want to cut the restriction endonuclease, the gene in between. I want to clone this fragment from here to here. There, unless there is a specific restriction site here, it's not useful. But CRISPR, we can use it as we want, because if you remember the CRISPR RNA, you can always design it. If I know this sequence, say for example, I know these two sequences, and I make a CRISPR RNA based on that, right? If I make CRISPR RNA, assume something like that, and Cas9 is provided, then it would, the cleavage would have occurred only here. Then I could get, not that people clone like this, it's not that easy to perform all these things in vitro. But the important thing is CRISPR RNA will give us flexibility. We don't have to wait for, or we don't have to search for a restriction endonuclease and hope that there is a restriction endonuclease exactly on these sides like that. Instead, we can design the sequence where it should be. There is limitation about PAM sequence and others, but still it provides certain flexibility. Okay. I will just finish it in 2 minutes. So, but we have 3 components now for cutting to be made. We need a CRISPR RNA, we need tracer RNA, and we need the Cas9. So if we have to clone these and make them, we'll need 3 different genes, and then hope that this base pairing between CRISPR, when they are produced, the base pairing between the CRISPR and tracer RNA would happen properly. Instead, genetic manipulation has started here. They made it this continuous like this by adding a linker loop like this. Now, how many components are there? This is the, in this case, 5' and 3' CRISPR RNA. Tracer, we have 5' and 3'. Now what they did, they added a linker loop like this. Now it is one single-stranded RNA, which is together, which is a composite of CRISPR RNA as well as the tracer RNA. And that is called as single guide RNA, right? If we want to now perform any cleavage, assume we only need 2 genes. One gene for the SG RNA, SG is for single guide RNA, and the other one is for Cas9, right? We will have more efficient control of what is happening then. All we need to do now is change this sequence based on where we want to cleave it. But of course, we can only, for recognition of CRISPR Cas9 complex, the target site should always be near a PAM sequence. Okay, remember first time how it is acquired? Cas9, Cas1 and 2, they would only cleave where there is PAM sequence, and then that region is taken as the protospacer. So the association with the PAM sequence is a part of the Cas9 protein for activity. So that is one important consideration we should have. Okay, this will be the last slide comparing this natural CRISPR pathway and the engineered one. In natural CRISPR-Cas pathway, it will produce tracer RNA, and it will produce the CRISPR RNA from the pre-CRISPR, and it will it will bind with the Cas9, and it is active. When it finds a target spacer within adjacent to a PAM sequence, then it will cleave it. Whereas in engineered one, we have a sgRNA gene which is single guide RNA. There is a gene for Cas9. This is a plasmid. Nowadays it's very simple to do as well. The expression of it will produce the single guide RNA, which will form a complex like this almost. And then when it binds with the Cas9, it becomes active Cas9 and ready to cleave the target sites. Okay. Do you have any questions? In the next class, we will continue about editing and how it is useful, how it is applied. Okay. Do you have any questions?
No, sir.